This is the third talk of the Biocognizance lecture series, wherein we bring eminent biologists from all parts of the globe to share with us details of the areas of research and the work that they have undertaken or are undertaking in their respective areas. Before formally introducing today's speaker, I would like to go over a few housekeeping rules very quickly. Please keep your microphones muted. In case you have any questions, feel free to use the chat box to direct any questions that you may have to us, the moderators of today. We'll publish the questions for everyone thereafter, and we'll place them to the speaker at the end of the talk. I would now like to introduce the speaker of today's session. We are extremely privileged and honored to have with us today Professor Robert Weinberg, the director of the MIT Ludwig Center for Molecular Oncology, the Daniel K. Ludwig Professor for Cancer Research at the MIT, and the American Cancer Society Research Professor. Professor Weinberg, who received his PhD in biology from the MIT in 1969, received postdoctoral training in the Weizmann Institute and the Salk Institute. In 1982, Professor Weinberg joined the faculty as a professor of biology and at MIT, and he helped found the Whitehead Institute. He published his landmark paper, Mechanism of Activation of a Human Oncogene, in the journal Nature in '82. In 2000, he co-authored a seminal paper with Douglas Hanahan titled The Hallmarks of Cancer, which enlisted 6 requirements for one renegade cell to develop into cancer. He is also the author of the classic textbook in cancer biology, which is The Biology of Cancer, as well as two other books intended for a wider audience. One, Renegade Cell, How Cancer Begins, and Racing to the Beginning of the Road, The Search for the Origin of Cancer. Professor Weinberg is the discoverer of the first human oncogene RAS and also the first cell tumor suppressor protein RB. He has been bestowed with numerous awards in his life, which include the National Medal of Science, the Wolf Prize in Medicine in 2004, the Breakthrough Prize in Life Sciences, to name a few. His laboratory investigates 3 broad questions related to the origin and spread of cancer. First, how do cancer cells within a primary tumor acquire the ability to invade and metastasize? Second, how are the stem cell state and the epithelial-mesenchymal transition interrelated? And third, how are the regulators of epithelial-mesenchymal transition able to activate this profound change in cell phenotype? With this brief introduction, I would now like to request Professor Bob Weinberg to deliver the talk to the audience.
Thank you very much, Mr. Chakraborty. I would say hello to everyone. Namaste, namaskar. And we'll begin to move into my lecture on the malignant progression of carcinoma cells. When we look at how an individual tumor develops, in this case, in the context of the colon, we see a whole series of histopathological stages in which a normal tissue evolves progressively into more and more malignant cells. This is a step of multi-step tumorigenesis, a form thereof. And we know that in a whole series of different types of tissues, we have these multiple steps that lead from normal to tumor pathogenesis. Just to confirm, is my PowerPoint coming across properly?
Yes, it's fine for us. It's fine.
Yeah.
Great. In the context of the colon, multi-step process was formalized in 1989 by Vogelstein and colleagues, in which they found that as cancer cells developed into an increasingly, or pre-neoplastic cells evolved into an increasingly aberrant tissue, they acquired a whole series of distinct genetic alterations, which were associated with and presumed to drive the process of multi-step evolution. Indeed, in my own laboratory, we found that if one introduced a whole series of distinct genes concomitantly into a normal human cell, one could transform these genes, and this allowed one to at least establish a mechanistic rationale for why one needs so many different mutant alleles in order to create a primary tumor. So the question is then, can we understand the pathogenesis, the development of a tumor, in terms of a whole series of somatic mutations that are accumulated progressively as normal cells evolve more and more towards a stage of first primary tumor formation, indicated here as carcinoma, and then ultimately invasion and metastasis. And this also raises the question of how important are non-genetic programs in determining the biology. If we look at one major factor that is non-genetic in determining the behavior of a cancer cell, here we see a situation where two tumors were evolved through, or developed through transgenic methodology in the context of the transgenic mouse model. And what one found was that these two tumors that were generated, that were genetically identical, albeit derived from slightly different cell types in the lung, actually had very different gene expression programs, which emphasizes to us the fact that the gene expression program of a tumor, and thus of a cancer cell, is influenced in part by the differentiation program of the normal cell of origin that pre-exists prior to the initiation of multi-step tumorigenesis. And indeed, we can imagine that this differentiation program that pre-exists in the normal epithelium continues to be expressed in and to influence the behavior of the cancer cells that have progressed to various stages of primary tumor formation. Here again, carcinoma representing a primary tumor. That having been said, one can also ask the question, what happens at the end of this process? i.e., how does a carcinoma cell progress and invade and metastasize? How does it disseminate to distant sites in the body? And here, once again, I will present evidence that there are non-genetic mechanisms at work, that is to say, epigenetic mechanisms. This last step that I've just described, going from a primary tumor to metastatic dissemination, has often been described as the invasion metastasis cascade, and itself involves a whole series of distinct cell biological steps that enable cells in the primary tumor to ultimately enter into distant tissues and form metastatic colonies, which represent the sources of the mortality of 90% of human cancers. And obviously this complexity raises the question of how cancer cells acquire all of these distinct cell biological traits. Do cancer cells need to acquire additional somatic mutations in order to move from primary tumors, invade and metastasize? Or is the last series of steps dependent on non-somatic mutational mechanisms, on epigenetic mechanisms that do not involve changes in the sequence of the genome? And one answer to this is provided by the cell biological program that is illustrated here. Here is our human breast cancer cells. Being breast cancer cells, they start out epithelial and express an epithelial marker, seen here, cytokeratin in the red. Outside in these implanted tumors are the stromal cells, in this case of a mouse host. But of great interest to us here are the green cells, because the green cells derive from the red cells. They have shut down the pre-existing epithelial marker, cytokeratin, and they now begin to express instead Vimentin, which is a mesenchymal marker, and that suggests they've moved from an epithelial to a mesenchymal phenotypic state. That can be seen here as well. Here's a tongue of carcinoma cells. Initially, they express cytokeratins, rendering them epithelial, but you'll see here on the outside of this tongue of carcinoma cells, adjacent to the surrounding mouse stroma, you'll see cells that have shut down the epithelial marker cytokeratin, and instead now begin to express the mesenchymal marker Vimentin. They've moved from an epithelial to a mesenchymal state, a shift that is called the epithelial-mesenchymal transition. It's a complex cell biological program. And from the last slide, you can begin to understand that here, the position of these cells in direct juxtaposition with the stroma determines the shift in their cell biological phenotype, not a mutational change. This was first described by Elizabeth Hay, this entire cell biological program. I show you here a whole series of distinct phenotypes which are changed upon a cell activating this previously silent or latent cell biological program. Many of the traits that are acquired during the activation of the EMT program, we begin to associate with advanced malignancy, that is the cells of malignant carcinoma cells. And here's one example of this. Here's a carcinoma cell that is existing adjacent to a stromal cell indicated over here. And what one can see here in terms of the biochemistry of signaling is that the carcinoma cell initiates a series of interactions that begins through its release of interleukin-1. That in turn impinges on the cells in the stroma that express the interleukin-1 receptor, and the latter cells in the stroma then reciprocate by sending signals back to the carcinoma cell that in turn initiate within the carcinoma cell the activation of the EMT program. In other words, this reciprocal communication, paracrine communication, causes the carcinoma cell to turn on this previously latent program, which we saw before in the context of the micrograph. In fact, there are many alternative EMT programs, epithelial-mesenchymal transition programs. They share in common many of these traits that I showed you before, but each of them has its own idiosyncratic, peculiar variations on the theme. It is not as if there's always one rigid canonical EMT program. There are variations on this theme. We should realize that the EMT program was already evolved very early in Metazoan evolution, already in the Cambrian or the Precambrian, and a series of transcription factors whose names are given here are responsible for activating the EMT program. You see it in the dark areas in these embryos, and these transcription factors were already present at the beginning of the Metazoan radiation, and they are conserved in all modern existing metazoans, independent of whatever phylum they work in. And so here we see in Drosophila one factors that's involved in gastrulation, but we express the same transcription in our cells, and each of these transcription factors is capable of inducing or orchestrating components of the EMT program. So the EMT program is important in various stages of embryogenesis and has been retained in highly conserved form over the last 600 million years, with all of these transcription factors being conserved in the genomes of all metazoa. Shifting gears momentarily, I would describe another aspect of this entire problem, because as was already demonstrated in 2004, one can actually fractionate the cells within a human carcinoma using fluorescent activated cell sorting, using these two cell surface markers into minority populations and majority populations. The cells in these two populations have identical genetic alterations. They share the same genome, but they reside in distinct phenotypic states. And importantly, as El-Hajj and Clark showed in 2004, the cells up here, when they are implanted within a mouse host, are capable of seeding a new tumor, whereas conversely, the cells down here are incapable of seeding a new tumor. As a consequence, they were called cancer stem cells because of this tumor initiating capability. And when one began to recognize that a hierarchical scheme illustrated here that is associated with the operations of and the organization of most actively developing tissues can actually be applied to this phenomenology of cancer. Here we see this hierarchical scheme in which a self-renewing stem cell is able to renew itself, but generate more differentiated progeny, where the stem cell continues to maintain its representation within the tissue. And the same kind of organization could be ascribed to cancer cells and tumors, where this is the tumor initiating cell which can trigger the formation of a new tumor, whereas the great majority of the cells in a normal tissue, but in this case in a tumor, are incapable of seeding a new tumor because they've undergone a measure of differentiation, obviously an epigenetic shift rather than a change in their genomes. And a question that was posed in my laboratory already a dozen years ago is the following. Is there any connection between the EMT program, on the one hand, which I described before, and the stem cell program that I have also just described, the work being started by Senduraimani from, by the way, Tamil Nadu. And so he asked the question, if one introduces into cells the two EMT-inducing transcription factors, in this case, snail or twist, can one actually alter the position of cells vis-a-vis their representation as non-stem cells or stem cells? In other words, by inducing the EMT, can one alter the representation or increase the representation of stem cells? Here we're talking about two distinct cell biological programs, the EMT program on the one hand, and the stem cell program on the other hand. And here we see the experiments that Sendo Raimani and Wenjun Guo did already in 2008, where they took normal epithelial cells and they introduced into these normal epithelial cells either the snail or the twist EMT-inducing transcription factor. And in so doing, they took a population of cells that looked like this. Here's the non-stem cells, here are the stem cells. And by forcing these cells to go through an EMT through the auspices of either snail or twist, they moved all the cells from a non-stem cell into a stem cell position, which provided the first indication that there is indeed a connection between the EMT program on the one hand and the stem cell program on the other hand. The precise reasons for this we still don't understand with any clarity. Here is an extension of this work in which one attempts to find connections between the EMT and stem cells, in this case, not in the context of breast cancers, but in the context of the normal mouse mammary gland. And here down, we see a cross-section of a normal mouse mammary duct. It's a bilayered epithelium. And what we see here are the luminal cells on the inside, the basal cells on the outside. We know that the stem cells in the normal mammary duct, not in a breast cancer, contain the normal mammary stem cells in this basal layer. And what we see here is that in the normal mammary gland, the slug, EMT-inducing transcription factor, is constitutively expressed in the basal cells in their resting state. And so the EMT in this case is not a idiosyncrasy, a peculiarity of the neoplastic stage. It also operates in a normal, unperturbed tissue. And indeed, in this work here, Wenjun Guo, he found that the slug EMT-inducing transcription factor is overexpressed by a factor of 100, more than 100, in the basal cells of the normal mammary duct relative to the luminal cells on the inside. And he could examine whether actually slug played a causal role in the EMT process. Was it more than just associated or correlated with the EMT program and the stem cell program? And to do so, he took a normal mammary stromal fat pad, indicated here, leaving it in situ, removing the incipient mammary ductal tree cells here, creating a cleared mammary stromal fat pad, and introduced into these cleared mammary stromal fat pads mammary epithelial cells. And it's known from work already 50 years old that if the introduced mammary epithelial cells contain mammary epithelial stem cells, they will seed the formation of an entire mammary duct, which is histopathologically and functionally indistinguishable from a normally generated mammary ductal tree. And so this is what the normal mammary duct looks like in the mouse mammary gland. And one can reconstitute this simply by implanting mammary epithelial cells in the cleared mammary stromal fat pad. And what Wenjun Guo demonstrated was that if one implanted 10,000 control mammary epithelial cells into a cleared mammary stromal fat pad, nothing happened. But if one took those cells and exposed them transiently to the slug and SOX9 EMT-inducing transcription factors prior to this implantation, implanted the cells, and then waited 6 weeks later, one could see now more than 100-fold increase in the representation of the normal mammary stem cells, whose existence was made visualizable through their ability to seed the formation of an entire mammary ductal tree, proving in a direct and rigorous fashion the association between the EMT program, and in this case represented by slug, on the one hand, and a mammary ductal morphogenesis and normal mammary stemness. In fact, SLUG, this EMT-inducing transcription factor, is found in the basal layers of a whole series of stratified epithelia throughout the body, but we have no idea what happens in simple epithelia in various tissues throughout the body, where we have no indication that SLUG is playing a role. So we don't know the universality, how widely applicable, generalizable this association of the EMT is with the stem cell state in a whole series of tissues throughout the body, which are single layered epithelia rather than double or multiple layered epithelia. Having said this, in the context of the pathogenesis of breast cancer in the context of a mouse, what was found by Xinye was that if one looked at snail high cells that express this EMT-inducing transcription factor, 1 in 91 of the snail high mammary carcinoma cells could seed a new tumor. That is, the tumor-initiating cells were in this case represented by almost 1%, more than 1% of the cells in the tumor mass. Whereas the cells that expressed another EMT-inducing transcription factor, which I've just associated with the normal mammary stem cells, here there was an 800-fold lower representation of stem cells, indicating in this case that there was a strong association between the cancer stem cells, the snail-high cancer stem cells, and tumor-initiating capability, which further reinforces the association between the EMT program and the stem cell program. And that can be reinforced even further through the following experiment. Here, one took a line of human breast cancer cells. These are called MCF7 RAS cells. They're known to be non-malignant. And so if one grows them in a mouse, one sees that the resulting primary tumors after 6 or 8 weeks have a smooth outer edge and in fact are non-metastatic. If, however, one takes these cells and prior to introducing them into a mouse, one drives the expression experimentally of these two EMT-inducing transcription factors and then shuts off these transcription factors and then implants the cells, now one sees that the resulting cells, 10 weeks later, for example, now have a highly invasive edge, as you can see here. They no longer behave like benign cancer cells. So they remember their prior exposure to the EMT program. And moreover, and highly interesting, is the result of their ability to metastasize. Because these carcinoma cells, if they're implanted on their own, normally form no micrometastases or macroscopic metastases in the lung, as you can see over here. If, on the other hand, they're exposed transiently to these two EMT-inducing transcription factors, now one can see as many as 400 micrometastases, they having metastasized from a site of subcutaneous implantation to the lungs, and about 100 macroscopic metastases, indicating that the EMT program can play a causal role in the metastatic process. And so this begins on its own to prove that by activating the EMT program, in this case experimentally, one can cause primary carcinoma cells, indicated here, to complete many of the steps of the invasion metastasis cascade that I described before. Indeed, it's highly plausible on the basis of what I've told you that simply by turning on the EMT program, one can actually force primary carcinoma cells to physically disseminate, to invade and metastasize. And it is my belief on the basis of this that the EMT program, when expressed in primary carcinoma cells, can allow them to physically disseminate and to form micrometastases in distant organs. The final step, which is here called colonization, involves by necessity the adaptation of carcinoma cells to living in a foreign tissue microenvironment, which is quite different from the microenvironment in which they grew up in the context of the primary tumor. And this requires adaptation. And to my mind, this adaptation, which the disseminated carcinoma cells need to undertake in order to thrive in a distant and foreign tissue microenvironment, such adaptation, I believe, cannot be under the aegis of the EMT program. So this is still, to my mind, an unanswered question. How do cancer cells learn to make a living in an unfamiliar microenvironment, such success being often termed colonization. Indeed, we know that there are strong proclivities of primary cancer cells to metastasize to distant organs at different rates. Here we see the fact that the breast cancer likes to metastasize to the bone marrow and to the lungs, but not to other tissues. And these differences in the metastatic tropism of these various primary carcinomas presumably reflect the ease or difficulty that disseminating cancer cells have to adapt themselves and to colonize distant tissues. What more can we begin to say about the EMT program? You might have assumed on the basis of what I told you until now that the EMT program causes epithelial cells to become mesenchymal cells. But the truth is that somewhere in the middle exist a whole series of intermediate states. And these intermediate states are partially epithelial and partially mesenchymal. They are not, in other words, the EMT program does not operate as a binary switch that flips cells from an epithelial state all the way over to a mesenchymal state. More often than not, as I'll emphasize momentarily, cells are pushed into quasi-mesenchymal states where they continue to retain certain epithelial characteristics, but have acquired some mesenchymal characteristics. And clearly along this spectrum arises the question, where do cancer stem cells reside along this spectrum from an epithelial phenotype all the way to mesenchymal phenotype? Do they reside somewhere here in the middle where they have mix of epithelial and mesenchymal characteristics? And we know that such a mix exists in real tumors, because here we see an actual xenograft tumor where there's a whole mixture of epithelial and mesenchymal cells, and some of the cells, without going into detail, co-express both epithelial and mesenchymal phenotypes, markers, which already begins to underscore the notion that one can have cells of mixed phenotype halfway in between the epithelial and the mesenchymal state. And this would allow us to begin to understand how epithelial and mesenchymal characteristics influence the behavior of carcinoma cells. In fact, here, as the work of Brian Beery showed, if one starts here at an epithelial level, and here over here is the extreme mesenchymal level, one can see that as cells progress from the epithelial state progressively more and more toward the mesenchymal state, one sees that they start shutting down E-cadherin, they turn on this cell surface marker, they turn on this mesenchymal marker, they shut down this pre-existing stem cell marker and shut down integrin beta-4 and turn on fibronectin. So there are different degrees of acquisition of the mesenchymal state. And as we and others have found, the cancer stem cells that I described to you before, and which are measured empirically, experimentally, through their ability to seed new tumors when they are extracted from a tumor and implanted into a mouse host, these tumor-initiating cells, abbreviated here TIC, these tumor-initiating cells reside in the middle. They're neither epithelial nor mesenchymal, but instead are quasi-mesenchymal. They're sitting here in the middle. In truth, if the EMT program gets activated in carcinoma cells, cancer cells rarely, if ever, move all the way over to the mesenchymal state, but instead almost always invariably end up in the middle, somewhere halfway in between. And if one measures the tumor-initiating cell frequency of mixed populations of cells, the cells that sit here in the middle, in this particular experiment, 1 in 3,000 cells had the ability to seed a new tumor. Whereas if one looked at highly mesenchymal cells, 1 in 750,000 were able to seed a new tumor. And this indicated on its own a 250-fold difference in the respective abilities of these different tumors to initiate or to seed the formation of new tumors. The carcinoma cells here are 250-fold enriched here than vis-a-vis with respect to the mesenchymal cells all the way over here. So this is indeed the place where the tumor-initiating cells, the cancer stem cells, reside along the epithelial mesenchymal spectrum. And one can also ask the question, are there discrete states along this spectrum. We don't really understand that very well yet. Do cells stop at particular stopping points, resting points along the road from epithelial to mesenchymal? And the beginnings of this can be understood in terms of a single cell RNA sequencing, which suggests that there are clusters of cells with specific transcriptomes that reside to differing extents along the epithelial to mesenchymal spectrum. That having been said, I wanted to pass to a specific consequence of the activation of the EMT program. And it poses the following question. Here I describe the work of Anushka Dongre, and it poses the following question. If we deal with epithelial and mesenchymal cancer cells, does their residence within one or the other phenotypic state affect their responsiveness to different kinds of immunotherapy? Because as you know, cancer cells have been increasingly treated with checkpoint immunotherapy as a way of trying to eliminate them rather than using chemotherapy. In other words, people have had some success in actually treating human tumors with checkpoint immunotherapy. And here, one of the most important of these checkpoint immunotherapy treatments involves the use of anti-CTLA-4 serum, because the anti-CTLA-4 serum blocks a negative feedback mechanism which shuts down the attack by a tumor-specific T cell. And in the presence of anti-CTLA-4 serum, one can then actually activate and unleash the previously silent, compromised ability of a tumor-specific T cell to attack an antigen-presenting cancer cell. So this represents a very powerful way of empowering the immune system in order to allow it to actually attack and eliminate a cancer cell. And the use of this anti-CTLA-4 serum then induces a reinvigoration of the functionally previously exhausted T cells. It requires the existence of T cells within a tumor. And in fact, we know from other work that tumors that lack any pre-existing T cell may not actually be benefited by using this kind of therapy. So one needs T cells within a tumor. They may be exhausted prior to the therapy, but if one uses anti-CTLA-4 antibody, one can actually invigorate the previously exhausted, previously functionally silent T cells, enabling them, finally unleashing them to attack carcinoma cells that are close by. Here is just a summary of various types of different kinds of immunotherapy as these immunotherapies have been applied in the clinic. And here one sees, for example, in the context of metastatic melanoma, which is unusual because it's so responsive to checkpoint immunotherapy, that normally the survival of patients having metastatic melanoma, in the case of chemotherapy or low molecular weight drug therapy, these individuals survive only a very short period of time using existing chemotherapies. But in the case of metastatic melanoma, if one uses two distinct kind of checkpoint immunotherapy, here you can see the origins of these two antibodies, then one can actually have patients which 20% of them may die relatively quickly. But here one sees in these metastatic melanoma patients that have been treated with these two distinct kinds of checkpoint immunotherapy treated concomitantly, that such patients survive for a long period of time and may indeed be cured. Curves like this one do indeed suggest a definitive cure of patients who otherwise, as you can see from this curve, would have been doomed to a rather quick and rapid death because of this aggressive disease. And here is a summary, for example, of how different kinds of tumors respond. And the truth is that this metastatic melanoma is very responsive to checkpoint immunotherapy, but many other kinds of cancer have not been so responsive. And that in turn raises the question of why these other kinds of carcinoma cells have not been responsive to checkpoint immunotherapy, in contrast to the responsive metastatic melanoma to checkpoint immunotherapy. And in order to deal with this, Anushka Dongre has created different cell lines that were prepared initially by Xinye. These are cell lines that are created in a transgenic model of a mammary adenocarcinoma pathogenesis in a transgenic mouse. And here one sees these cell lines that were derived from the tumors arising in these mice. One cell line is epithelial, and the other cell line is mesenchymal. Here's another pair of cell lines. One is epithelial, the other is mesenchymal. And one knows about these characteristics because the epithelial cells express E-cadherin, the hallmark keystone epithelial marker, whereas in contrast, the mesenchymal cells express a whole series of EMT-inducing transcription factors and these two mesenchymal markers. Interestingly, and for reasons that we do not understand, the epithelial cells are stably established in an epithelial state and do not lose this phenotype when they are growing in vivo in a tumor. Similarly, the mesenchymal cells are stably residing in the mesenchymal state, and once again, they do not lose this phenotype when they are passaged in vivo. You might think that because there is phenotypic plasticity, here I refer to the ability of these cells to interconvert from one phenotypic state to the other, back and forth, that might undermine this stability. But in contrast to one's expectations, the epithelial cells, as I've said, are stably epithelial. The mesenchymal cells reside stably in the mesenchymal state. And so what is the consequence of the immunological state of these two different kinds of carcinoma cells? Individuals, researchers studying the checkpoint immunotherapy have looked at the various kinds of immunocytes, immune cells, in the context of different kinds of human tumors. But few have actually examined what the consequences are of the residence of the carcinoma cells in one state or the other state, in the epithelial versus the mesenchymal state. And here we see some rather dramatic differences in the biology of these two tumors with respect to the immune cells that are present in the stroma of epithelial versus mesenchymal tumors. Here we see that if one looks at one pair of cell lines with a double-headed arrow, or another set of cell lines, pair of cell lines, once again matched with double-headed arrows, here we see that the epithelial cells have far fewer immunosuppressive macrophages than do the corresponding mesenchymal cells. In other words, these macrophages are present in far higher numbers in the mesenchymal tumors relative to the epithelial tumors. And here one can demonstrate that the macrophages that are present in the stroma of the mesenchymal tumors are present. These are immunosuppressive macrophages, which means that they help to blunt the attack on the tumor by the cytotoxic T cells. And these immunosuppressive macrophages are present in far higher numbers, in amounts, in the mesenchymal tumors than they are in the corresponding epithelial tumors. And from this, one already begins to get the impression that the mesenchymal tumors are able somehow to create an immunosuppressive microenvironment that might prevent their being eliminated by attacks from the immune system. Here's yet another example of this. If one compares the epithelial to the mesenchymal tumors, one sees that the CD8 positive cytotoxic T cells, these being the T cells that are responsible for recognizing and attacking targeted T cells, targeted cancer cells, these cytotoxic T cells are present in far higher numbers in the epithelial tumors than the mesenchymal tumors. So to the extent that the tumors would be eliminated by these cytotoxic T cells, which recognize and attack and kill the carcinoma cells, there's far more of these cytotoxic T cells in the epithelial tumors with respect to the mesenchymal tumors. And indeed, if one looks at the state of functional activation of the CD8 positive T cells, i.e. whether the CD8 positive T cells are functionally activated or are exhausted, that is to say, incapable of undertaking any biological attack. One finds that the carcinoma cells in the epithelial state are functionally activated here in the black, in contrast to their state in the mesenchymal tumors, where one sees a lot of exhausted T cells that are incapable of launching an attack. And here one can see this even more dramatically, where in fact, the representation of T cell exhaustion indicated here is far higher in the mesenchymal tumors than it is in the epithelial tumors. And together this yields the impression that the mesenchymal tumors are able to create an immunosuppressed microenvironment, an immunosuppressed stroma, which may protect them, for example, from attack by the immune system. In this case, the attack being launched by the CD8+ cytotoxic T cells. Here's yet another manifestation of this dramatic difference in the immune landscape of these tumors. You may know that a class of T lymphocytes called regulatory T cells, defined by the expression of these two markers, are strongly immunosuppressive, and these regulatory T cells suppress the activity, the ability of cytotoxic T cells to actually kill the cancer cells. And one sees that these immunosuppressive regulatory T cells are present at far higher levels in the mesenchymal tumors than they are in the epithelial tumors. And when I talk about epithelial mesenchymal tumors, throughout this, I'm referring to the fact that one takes the aforementioned epithelial carcinoma cells, implants them in a mouse, and observes that the resulting tumors remain epithelial, in contrast to implanting mesenchymal carcinoma cells and observing that the resulting tumors remain phenotypically mesenchymal. And here, yet even further contrasts, I'll just focus here on the micrographs, which I trust you can see in faraway Bangalore, where one sees that these red cells here are macrophages that are immunosuppressive, and these immunosuppressive red stained macrophages are present in far higher numbers in the mesenchymal tumors than they are in the corresponding epithelial tumors. Indeed, what is interesting is that the M2 immunosuppressive macrophages, so these macrophages prevent the immune system from launching attacks on the tumor. These immunosuppressive macrophages are exiled to the outside of epithelial tumor masses. They're not allowed, they're not permitted to enter. Whereas in contrast, in the mesenchymal tumors, there are large numbers of these immunosuppressive cytotoxic T cells, of these M2 macrophages. These immunosuppressive macrophages have invaded into the mesenchymal tumors, where they are presumably suppressing any attacks by the immune system on these tumors as a whole. And there are yet other differences. For example, as you may know, our cells throughout the body express the hands of MHC class I molecules. These MHC class I molecules are expressed on the cell surface. They carry in their hands, in their palms, oligopeptides that represent proteins that are present within the cells, and they provide to the immune system an ability to get what kind of proteins are actually being expressed in the cell. And what one sees here is that epithelial cells express far higher levels of these MHC class I molecules than mesenchymal cells. And the comparisons are actually quite dramatic. Here, if one turns on the EMT program in vitro, one sees more than 100-fold difference in the MHC expression, where the epithelial cells are expressing on their surface these MHC class I molecules, thereby enabling the immune system to see what's going on inside them by expressing intracellular oligopeptides on the surface. But the mesenchymal cells no longer express significant amounts of MHC class I on their surface. In other words, the mesenchymal cells are flying under the immunological radar because they're not expressing on their surface these MHC class I molecules, whose display on the cell surface is critical to the ability of the immune system to sense what's going on inside the cell or to sense what kinds of proteins are being made inside the cell. And so here are the results of attempting to use an anti-CTLA-4 checkpoint immunotherapy. I showed you in cartoon form before how it works. And here's an initial result of Anushka Dongre. Here is an epithelial tumor. These are carcinoma cells that reside stably in the epithelial state, and it grows out quite well. But if one applies the anti-CTLA-4 checkpoint immunotherapy, this tumor is kept nicely under control. Not totally eliminated, but kept nicely under control. Now it's about 80% reduced in overall mass compared with the untreated control. Here, quite dramatically, is the behavior of the tumors that are formed by the implantation of the more mesenchymal cells. And these mesenchymal tumors, whether or not anti-CTLA-4 checkpoint immunotherapy is being applied, they are totally resistant to being shut down by the anti-CTLA-4 checkpoint immunotherapy, which already tells us in ways that were hinted for before, that in fact these two kinds of tumors behave in a dramatically different way with respect to applied checkpoint immunotherapy. Epithelial cells are very sensitive, as you can see here, and mesenchymal tumors are totally refractory, resistant. Anushka Dongre then undertook an interesting experiment. She mixed together epithelial and mesenchymal carcinoma cells and asked how the resulting mixed tumors would behave. I would mention that she started out here with tumors in which there was a 10% representation of implanted mesenchymal cells and a 90% implanted commingled epithelial cells. So the epithelial cells were present in great majority, and this 9-to-1 ratio of epithelial to mesenchymal remained in the resulting tumors that grew out. In other words, the ratio between these two cells did not change. Here, I'm just showing you once again how a fully epithelial tumor behaves with respect to anti-CTLA-4 checkpoint immunotherapy. Here, I'm showing you once again how the mesenchymal tumors behave, showing you the earlier curves. And here's the interesting curve, because here in this particular set of tumors, there is a 9-to-1, there are tumors that are formed by a 9-to-1 admixture, 90% epithelial cells, 10% mesenchymal cells, and these cells behave quite similar to the tumors that are fully mesenchymal. And this yields a very interesting insight, and that is that the 10% minoritization of mesenchymal cells within a tumor can protect the 90% majority epithelial neighbors from immune attack. And this is of some consequence to actual human tumors, because we believe that most carcinoma cells arising in humans have minority populations of mesenchymal carcinoma cells. And if they exist indeed, these minority populations may actually protect the tumors as a whole. In other words, that the tumors as a whole may be quite resistant to attack simply because they harbor minority populations of mesenchymal cells that protect the tumor as a whole, the mesenchymal cells conferring cross-protection on their epithelial neighbors. And actually, we don't understand how this works. Here's one of these mixed tumors I've been describing to you, and what you see here topologically is that the epithelial cells congregate with their own, and the mesenchymal cells congregate with their own. In other words, they do not intermingle, they do not integrate. Instead, they are segregated one from the other. And that begins to raise the puzzle of how the mesenchymal cells, which I described to you before, are able to protect their epithelial neighbors. This micrograph, by the way, misrepresents the actual overall proportions in these mixed tumors, because I've told you that the mesenchymal cells represent only 10% of the tumor and the epithelial cells are 90%, something you could not deduce from this particular micrograph. Importantly, in these mixed tumors, the epithelial cells continue to look epithelial, and the mesenchymal cells continue to look mesenchymal. And so they continue to preserve their pre-existing histopathology. And as Anushka Dongre grew up here, there is actually a profound difference in the appearance of these tumors with respect to the representation of various kinds of immune cells within these tumors. And here I'm just summarizing in cartoon form how these tumors are put together. As I told you before, the mixed tumors are actually quite immunosuppressed, as are the purely mesenchymal tumors. And the mixed tumors are immunosuppressed because the minority populations of mesenchymal cells are able to protect the tumors as a whole from attack by the cytotoxic T cells, which are illustrated in pink. Here's the epithelial tumor in which the cytotoxic T cells are active and are invited into the tumor, where they succeed in attacking and eliminating the previously existing epithelial carcinoma cells. So a quite different spectrum of cell types present in these different tumors. Anushka Dongre attempted to determine how can the mesenchymal carcinoma cells protect the epithelial neighbors. And in so doing, she converged on a series of 7 different secreted factors, actually 8 shown here, which are secreted proteins that are expressed by the carcinoma cells, the mesenchymal carcinoma cells. These 7 here to the right being various kinds of secreted cytokines. And this begins to indicate that to the extent that mesenchymal cells can protect their epithelial neighbors, they do so by releasing or displaying a whole series of proteins that allow paracrine signaling between the mesenchymal minority and the epithelial majority in these cells. And here is a listing of these different proteins which the mesenchymal carcinoma cells seem to display and release in order to, so they can deploy these various factors to protect their more epithelial neighbors. And what she has found is that if she knocks out some of these, what she finds, to make a long story short, is that if one knocks out either in the mesenchymal carcinoma cells, either CD73 using CRISPR-Cas9, or one knocks out in the mesenchymal carcinoma cells CSF1, or one knocks out the gene in the mesenchymal carcinoma cells for SPP1, then one begins to see something very interesting about the cytotoxic T cells I've been describing previously. In the mesenchymal carcinoma cells on their own, there are no cytotoxic T cells, as indicated here by the absence of red cells. But if one knocks out either any one of these 3, one or the other, now one sees once again the reintroduction of cytotoxic T cells that are now permitted to enter into the tumors, where they presumably can launch various kinds of attacks on the carcinoma cells. And here one begins to see the work from Anushka Dongre about how these cells respond to checkpoint immunotherapy. Here are highly mesenchymal carcinoma cells, and with or without checkpoint immunotherapy, they behave identically. If one knocks out CSF1 in the genomes of the mesenchymal carcinoma cells, now there is a partial sensitization. If one knocks out SPP1 in the genomes of the carcinoma cells, there's also a small amount of sensitization of the carcinoma cells that were previously fully refractory to attack. But here's the dramatic result. In these carcinoma cells that were previously mesenchymal and were fully refractory, as I just showed you, now they can be eliminated by blocking the ability of the mesenchymal carcinoma cells to produce CD73, which is a cell surface protein that is displayed by them. And in the absence of that, these mesenchymal carcinoma cells present in the minority within the tumor can no longer protect the mesenchymal tumors from attack by cytotoxic T cells. And now one sees that these tumors are eliminated. So we begin to get a feeling for the dynamics, the physiological interactions between the mesenchymal and the epithelial carcinoma cells, which may be reflected actually in the biology of existing commonly occurring human tumors. And so here is a recent photo of members of my group. Actually, the photo you see here is in our cabin in the woods of New Hampshire. And I've been actually in this cabin for the last 7 months, living here in COVID exile, far away from the city, where there is much more virus and where one would like to avoid the dangers of being infected by individuals who are running around as loyal Trump supporters, not wearing masks. So on that note, I would like to thank you for your attention. I would like to thank Mr. Chakraborty and colleagues for the kind invitation to participate in this symposium. And of course, I'd be very willing to take a whole series of questions from you in the 10 minutes or so, or 13 minutes that, that we continue to have together. So
yes.
please.
Yes,
so
right. So we will try to take as many questions as we can in this time. Do tell me when you need to go. I'll definitely try to cut off before 7:35 here. So anyway, within like 10 minutes. Right, so
Perfect.
one of the questions asked is there appears to be a lot of overlap between cells undergoing EMT and cancer stem cell phenotype. For example, cancer stem cell specific transcription factors like snail and twist are upregulated in both types. The question is, are they the one and same? And if yes, cells undergoing EMT are actually cancer stem cells or reprogramming of differentiated bulk cells to cancer stem cells? And can all cancer stem cells metastasize if a chance is given?
So there's a whole series of questions here. What I was attempting to argue is that if one has fully epithelial cells, epithelial carcinoma cells, and one turns on a partial EMT program where one pushes cells from fully epithelial to a state to a quasi-mesenchymal state with mixed epithelial and mesenchymal traits, such cells have a far higher representation of cancer stem cells. However, unfortunately, we've never been able to isolate pure populations of cancer stem cells, even though we know that they reside somewhere in the epithelial to mesenchymal spectrum. We don't know exactly where. And this is an abiding mystery. We don't understand why the EMT program and the stem cell program are so intimately interrelated to one another. We don't understand why, when you turn on in fully epithelial cells a partial EMT program, you create far more stem cells than existed previously. I don't know whether that helps to respond to that question. I suspect it does. Please.
All right, so basically a lot of this is still under research, like as in we don't know.
Absolutely. Yes.
All right. So the next question is, it's kind of like a part. Actually, I think the previous question answered it. Are the similar stromal factors involved for transition to cancer stem cell types from non-stem cell, as well as in the case of EMT?
Well, I think the question is here twofold. First of all, how does the stroma influence the carcinoma cells? And we know that, to begin, the activation of the EMT program in previously epithelial carcinoma cells occurs because there are certain signals that are released by the stromal cells from the activated stroma, such as TGF-beta and canonical Wnt proteins, that impinge on the carcinoma cells. I showed you yet others before, and activate a previously latent EMT program in the carcinoma cells. Once the carcinoma cells in turn enter into a quasi-mesenchymal state, including within them some cancer stem cells, they respond by releasing signals that change the composition of the immune cells that have been present in the stroma. So there's a reciprocal back and forth signaling between the carcinoma cells and the stromal cells that they recruit. And when I say stromal cells, I include not only fibroblasts and myofibroblasts, but importantly, in the context of checkpoint immunotherapy, I also include in the stroma various kinds of immune cells.
All right. So another question is, could altered integrin repertoire on circulating tumor cells determine organotropism?
So this is a very interesting question of how different kinds of primary tumor cells end up growing preferentially in one target organ or the other. I'll give you my preconception, but it's only a preconception. And the preconception is carcinoma cells are released by the primary tumor and scattered all around the body to various kinds of tissues. They probably invade in various kinds of tissues. But in some tissue microenvironments, they're totally incapable of thriving, whereas in other tissue microenvironments, they begin to proliferate. That raises the question of what enables carcinoma cells to thrive in one tissue microenvironment and fail to adapt in yet another. And it could well be, as the questioner poses, that in fact the ability to thrive in certain tissue microenvironments may depend in part on the ability of the carcinoma cells to express certain cell surface integrins. That's a distinct possibilities that remains unexplored.
All right. So another attendee asks, the data shown with respect to mesenchymal tumors are with anti-CTLA4. Do mesenchymal tumors resist anti-PD-1 therapy? Will combination therapies work better with mesenchymal tumors?
So this is a very interesting question, and I would respond as following. It turns out that CTLA4 therapy, as I showed you, is very effective in eliminating anti-CTLA4 therapy is very effective in eliminating epithelial carcinoma cells. The PD-1, PD-L1 alternative form of checkpoint immunotherapy does not work at all in the context of the particular mammary carcinoma cells that I showed you here. This raises the question of whether the take-home lessons that I described today are applicable as well to the PD-1, PD-L1 form of checkpoint immunotherapy. At present, we can only conclude that these lessons are relevant when using the CTLA4 immunotherapy. And it remains to be determined why these particular mouse mammary carcinoma cells turn out to be unaffected by the PD-1, PD-L1 checkpoint immunotherapy. We don't understand it. So that's still a mystery, and it was a very good question posed by the listener.
All right. So what would be the outcome of co-injection of epithelial and mesenchymal cells on the immunosurveillance in the tumor microenvironment? Will the mesenchymal cell-influenced immunosuppression protect the neighboring epithelial cells? That is the question.
Well, my intent was to present the argument that one has this mixture of different carcinoma cells, that the mixture of different carcinoma cells allows the mesenchymal cells to dominate, even though they may be present as a small minority of the carcinoma cells, and they in turn can dictate how the immune microenvironment is in the stroma of the resulting tumor. Dictate in the sense that they can influence and create an immunosuppressive stroma, even though the epithelial carcinoma cells within the same tumor are present in great majority. Precisely how they do that, we don't really understand, but I gave you one clue, which is to say that the expression of CD73 on the cell surface of these cells, which causes the release, and which is known to be an immunosuppressive molecule, that represents one way by which the mesenchymal carcinoma cells are able to dictate the outcome of the interaction. Perhaps we'll take one more short question, and then I fear I have to fly off to Heidelberg to give another lecture, far away, without getting into a jet plane. Is there one final question? I can't hear you now.
So I guess you're muted.
Mute.
Yeah, gotcha. So quickly, is there a role of tumor stroma in epigenetically activating oncogenes which can further activate the EMT program?
I can't, I really can't answer that. We don't know much about that at present, unfortunately. Right. So on that note, I would like to say again, thank you for having me.
Yeah.
I don't know how to say it in Kannada. In Tamil, one says Nandri, Shukriya, Dhanyawat, and hope to meet you somewhere soon on this globe. It's been a pleasure to be with you, even though I'm about 8,000 miles away from you right now. So thank you again for your hospitality. I wish you all a good day. And may you survive the current COVID crisis, and may we all emerge from the COVID crisis and the presidency of Donald Trump in good shape.
All right. Thank you so
Good
much,
day to
Professor
you.
Weinberg. We'd like to thank you immensely for
And
taking the
bye-bye.
time. Yep. Thank you
It's been my
immensely for
pleasure,
taking
certainly,
the
without
time out of your day to give this talk. And yeah.
and I'm certainly flattered that there were so many participants that had joined us today. So, Mr. Chakrabarty, thank you again for organizing this, and I trust it helped.
Thanks a lot.
to
Thanks
enrich
a
your
lot to
support.
you for making out time for from your busy schedule to deliver this talk for all of us today. Thanks a lot.
This is how I make a living, so it's not something out of the ordinary for me. Again, many thanks. Stay healthy and continue in your studies. You can see outside behind me, we're in the middle of a storm here, and it's cold. Whereas in Bangalore, it may be warm and sunny, whatever. Okay, again, bye-bye, everyone.
Thank you. Bye.
Thank you, sir. Bye.
So we would further like to thank the Dean of the IASC UG program for making all of this possible. We would like to thank Team Pravega for their support in the organization of this talk. And lastly, we'd like to thank you, the audience, for attending this talk, and we sure hope you enjoyed it. If you did, Pravega is hosting a bunch of other talks, including the next one, which is on 17th, by Professor Malcolm H. Levitt on NMR spectroscopy. As for BioBlitz webinar series, we will be back soon, and stay safe and take care.
Yeah, at the end, I'd just like to add that we'll be releasing the recorded video of this talk today on our YouTube channel. So those of you who have missed the talk or wish to take a look at it again, so you may please look up in our YouTube channel. Thank you once again for attending this talk, and have a nice day.